Good evening, gentlemen. Welcome back to class. I hope you all had a fantastic Christmas break. We are this week going to be moving into Chapter 2, and there are 3 sections to this chapter. There'll be 3 sets of notes that you will be responsible for listening to these lectures and taking these notes during the course of your CQ time, especially in the first half of the week. Keep in mind that our schedule will be as follows. You will be prepping for your group oral quiz, which will take place on Thursday morning. And on Friday, you will have a Chapter 2 test that you will take on the computer, like your Chapter 1 test. During the course of this week, I'm sure we'll also be talking about making sure that we're prepped and ready for the end of the first quarter and the first quarter exam. And we'll be looking at some lab activities that we will be doing this week, as well as introducing the soil analysis project, which you'll be working on right up until the end of the course, week 8. But just keeping that stuff in mind, let's go ahead and prep ourselves and get ourselves ready here for Chapter 2. We have a list of different things that we're going to be covering here in this first section. We're going to spend some time looking at the scientific method and striving to understand it so that we can apply it properly. This means that we're going to break it down and look at the scientific method in terms of steps or parts, and then we'll have some other things that we will look at, not nearly as intensely. Take a look at this question here and think about this, think about how you would answer this, how do you use it, and we're going to kind of apply, figure out how to apply this in terms of the scientific method. Again, this list of questions here is a good list of questions for you to be thinking about in terms of the group oral quizzes in particular. Most of the stuff will be stuff that we go over in detail in the lecture. All right, the experimental method or the scientific method, you'll hear me use those terms interchangeably, so don't get confused by that. This is the method that scientists use to do their research. Generally, today we understand it as the proper method or the how in terms of how science is done. And if this is the process that scientists are using in order to do their research, then we recognize it as the process in which discoveries are made within science. And we're gonna break it down into a series of steps. And so it is important, guys, that we think about these steps in a particular order. And it's important for you to learn them in that order, but also more so, especially at this stage, for you to be able to do them in that right order. I will say that real, true science discovery or research is not cookbook science. By cookbook science, I mean, typically in a class where you do a lab where it gives you a set of steps and you do them in order and you get an outcome. I think that's good for learning skills in the lab, but out in the real science quote unquote world, research tends to be messy, and although a research scientist is going to want to think about these things in the experimental method in the correct order, doesn't necessarily mean that they do everything all the time in that order, because things happen and they have to be able to be flexible in order to continue the work. But it's important for us to to learn them in proper and appropriate order. The first is observing. In observing or making observations, we obtain information by using the senses. We also use the word observation to refer to the information that is obtained by using the senses. So you can think of it as a verb, or you can think of it as a noun. When we talk about the senses, we're talking about taste and touch and sight and smell and hearing, the 5 typical senses. Now, obviously, we are not always going to apply these in every scientific endeavor. Some are more appropriate than others. If you're in a lab, you generally don't taste things, okay? You generally don't smell things. In chemistry, there's an actual skill that we call wafting, where we try to get a slight whiff of what may be happening in the test tube, but we're always careful about these things. We generally aren't looking to put things into our body or ingest things, and so it's a little broad to talk about using all of the senses. Classically, in science, when we talk about the use of the senses to make observations, we're classically talking about sight and what we see, whether it's what we see with the naked eye or what we see magnified through a microscope or through a telescope. If we go into geology and earth science, there's some hardness tests for minerals where we're going to have the sense of touch that's in play. But I really, really want you guys to be thinking about this and think about science at its most basic and its most fundamental level. We gain understanding, we make discoveries based on what we can observe. And that's what's important here. And that's why it's the primary, or it's the first step of the scientific or the experimental method. And then we do things with those observations. We write up a description. If you're a naturalist and you're studying birds in an area, you're going to write in your journal as much as you can about that bird, as much as you can see, as much as you can take in. You're going to write down, you draw pictures. That's what scientists used to do. They used to draw pictures of everything they saw. I remember taking my biology classes even in college, and biology was really viewed as a class of drawing. You're drawing, making diagrams of everything that you see. Today we have the ability to take photographs, and if you can get really hands-on with what you're studying, you take measurements, and so you want to think about, and we'll talk about this, but you want to think about moving the observations into the realm of data and creating data points. But it starts with bare bones observations. From there we move on to hypothesizing and predicting, and I'm going to tell you guys right now, it's really, really, really important that you strive to understand what a hypothesis is, what predicting is, because one of the things that I have seen in my teaching of high school students is that there's a weakness in our explanation and therefore the student's understanding of what a hypothesis is, what predicting is, and that weakness ends up tending toward a weakness in understanding how to do science. Okay, so I would say that the observations that we make are the primary, the foundational thing that we do in the scientific method, but our hypothesizing and how we do it and understanding what a hypothesis is the most important step and skill in the scientific method. Good science, really good science, is tied to our understanding and therefore our application and the implications of what hypotheses are. Maybe some of you were able to get that out of that first movie we watched. We're going to talk about this. By definition, a hypothesis is a theory or an explanation that is based on observations that can be tested. So here again, guys, recognize this starts with the observations, but then we have to go down a propositional road. And what I mean by propositional is we have to take these observations, we need to put some things into words. And on top of this, we need to make sure that we are talking about things that can be tested. The science that we do in the classroom, the science that researchers do in terms of, we're trying to find a cure for cancer, we're trying to find an alternative energy source, that type of stuff, that level of research, it has to be in terms of what we can test, what we can see and what we can test. And when we get outside of those realms, we may still be doing science, but it's science of a different sort. And in today's world, not being able to differentiate these two and understand them can cause some real problems. Forming the hypothesis is the second step of the experimental method, and it's an explanation. Okay? So now I'm trying to explain in words what I saw in my observations. And I'm trying to explain what I saw in such a way that I can test it to see if my explanation is correct. Let me say that again, okay? I am now trying to explain in words what I saw in my observations, and I'm trying to explain it in such a way that I can test to see if my explanation is correct. So all the research that we do is based upon how good of a hypothesis we come up with. A hypothesis that is not worded in such a way that it can be tested is no hypothesis. So, if you've ever had people explain a hypothesis to you as an educated guess, that is entirely incorrect. I know that we even have textbooks here that we use that will describe a hypothesis as an educated guess. And that is just not the case at all. It's based on information that we already have. It's an explanation, the best explanation that we can come up with of what we have seen based on the information that we already had. That is a lot more than an educated guess. And I want you guys to be thinking about that and understanding that. It should make sense. It should be logical. It should be logical and make sense in that it follows from what we already know. So again, based on our observations, okay? If an example would be, in an understanding of why the sky is blue. Okay, think about it this way: we can look up into the sky, and we can see that it's blue on a clear day with no clouds. We can see a blue sky. We can see different colors in the morning during the sunrise, and we can see different colors in the afternoon and the evening during sunset. And if that's all we have, we don't have any other knowledge, that observation, that bit of information is all that we have. What's the best explanation that we can come up with why the sky is blue and why we see different colors in the morning and in the evening? And based on the observations, our explanation of that could include something about the sun and about light, and could maybe include something about the angle of the sun and the light, because we know those colors change in the morning at sunrise and in the evening at sunset when the angle is different in one sense than during the day. And now we're getting to having some sort of an explanation, and maybe maybe we can test it, okay? That is what a hypothesis is. And I want you guys to think about something. The better your hypothesis is, and the term I would use is maybe the tighter. Okay, the tighter your hypothesis is, or the better your explanation is, ultimately what that means is the better your test and your test results are going to be. Okay, a tight, real sound, really well-worded hypothesis is going to lead to a really good experimental design. And that's the type of stuff we want to be thinking about in science. Now, from hypothesizing, we come to predictions. And by definition, predictions are statements made in advance that express the results that will be obtained from testing a hypothesis if the hypothesis is supported. So I've got my observations. Based on my observations, I come up with an explanation of what is happening. Okay? That's your hypothesis. Really good, logical explanations of what is happening based on my observations. And then I make a prediction. And I say, if my hypothesis is true, then this will happen when I do my experiment. Now, predictions become really important when we start looking at the results of our experiments. We use the prediction to test the hypothesis. Somewhere along the lines, I had somebody teach me, I don't remember if it was in high school or in college, but somewhere along the lines I had somebody teach me a good hypothesis is written as an if-then statement. Okay? If blah, 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 then blah, 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 blah. And anymore, I don't think that is the best way to think about it. I think the if-then statement, the if-then construct, or the if-then formula is really how we make our prediction. The hypothesis is the really good logical explanation of what we saw. The if-then statement is the prediction. That if our explanation is true, then when we do something, we'll get this result. And so I hope you guys can see that how you design your experiment, how you do things, really is going to follow my initial observations. My hypothesis is really tight. I make my prediction, and then I go and design my experiment and see if my prediction was true or not. Now, you have to write these things, you have to think about these things in a sense that it has to be able to be disproved as well. We do not write hypotheses in such a way that they can only be proved. That is injecting bias into what we're doing. So one of the ways that we know we have a good hypothesis, and that it's tight, and that it's a strong explanation, one of the ways we know that is that the hypothesis can be disproved. And why this is important to science is because we are gaining knowledge. If we prove a hypothesis, we've gained knowledge. If we disprove a hypothesis, we've gained knowledge. This is where, you heard me talking in the first week about bias and keeping bias out of science. This is why this is important. Because if we are unbiased in our approach to wanting to gain more knowledge and more truth, then we are doing things in such a way that whether we prove or we disprove, we still gain knowledge. Okay? And even as it says right here, when we disprove things, we are reducing the number of explanations for our hypothesis. Or for our observations. Say we make some observations, write up our hypothesis, and we know that there's probably no more than 10 explanations for our observation. Well, if we set up our hypothesis, we do our experiment, and we disprove the first hypothesis, now we know that there's only 9. And we rewrite things and redo things and disprove the next one. We know there's only 8. We're still gaining knowledge in that endeavor. And you may think of it as working the problem backwards, but what ultimately a scientist is doing is zeroing in on what would be the remaining explanation that's left. Right, next, we do experiments. Okay? By definition, experiments are procedures that are carried out under controlled conditions to discover, demonstrate, or test a fact, theory, or general truth. So what's really important here, guys, is that in true science, we don't jump right into the experiment. In true science, we make observations, and then we try to explain those observations, and then we try to predict, and then we design or we make up the experiment based on that work that's already been done. And so a lot of mental work, a lot of thinking work, a lot of creative work, a lot of imagination work goes in before we ever get to the experiment. And I'll tell you guys something. I think in my opinion, one of the most challenging things about high school science is that we generally don't go through all of that thinking and all that creative and all that imagination and all that critical skills. We have a tendency, we teach a principle, and then we put a lab together that demonstrates that principle. Now I don't want you guys to get me wrong. There's absolute merit in that. If something is already a law in science, we're not trying to discover something new, but it's already a principle that's a law, then it's beneficial for the student to learn that law and then to see that law demonstrated in an experimental type setting. Okay, if you're going to go into science, there is a certain body content of knowledge that you should already have. On the other hand, I think what can happen is that we think that is the only way science is done, and science, there's a creativity to science that gets lost by only doing things that way. Experiments are not just found in a book somewhere that tells us what to do and how to do it. Most experiments were created from scratch by somebody somewhere along the line, and then once that experiment was able to show or to disprove what that person was working on, that experiment became a technique that all the other scientists had access to and could use for whatever they were testing or whatever they were looking at. When I worked in research, we read journal articles to see how somebody created an experiment to see if we could use their experiment as a technique in our lab. And then eventually things become a technique, and it trickles over time down even to like a high school science class. But experiments are important. You've made your observations, and you explain, and then you explain, and you explain, and eventually we get to a point where we can't come up with an explanation anymore. We've explained down to our last bit of observable knowledge, and that's when we put the experiments into place, and we use the experiments to try to even further our knowledge, let our explanation go a little bit further. That's why experimenting is important. And good experiments follow from good hypotheses, but good experiments are laser beam focused on cause and effect relationships. And we have certain tools that we use in our experiments to make sure that they're laser beam focused on cause and effect relationships. So we talk about variables. We talk about using variables and using controls, and that helps us focus the experiment. A variable is the factor that changes in an experiment in order to test the hypothesis. So you've got your hypothesis, you've got your explanation, you've got your prediction, now you're setting up your experiment, and you've got the thing you want to test. So that's the thing you're changing. If I do it this way, it doesn't have an effect on the outcome, as opposed to if I do it this way. Or I could do it 3 different ways. It's this one thing that I can change over and over and over and over again to see if it's going to have an effect. And that's the variable. And variables are really important. To test for one variable, scientists will study 2 groups or situations, the variable being the only difference between the 2 groups. So it's the one thing we're changing. Temperature can be a very common, very easy variable to change. You want to see, do these plants grow better at a average tropical temperature or an average Arctic temperature? Or you could make that variable and change it on a gradient. So you run 10 different groups all at the same time, and but at 10 different temperatures. So you see what we mean about changing, that's the variable. In that example, temperature is the variable. And what you would want to do at that point is you would want to try to set up your experiment so that you could control every other thing. You control amount of water, amount of moisture, you control amount of light. So the plants are getting the same exact, they all get the same plant food, same amounts, the same exact everything. 10 different cases, except each one of them gets a different temperature, and you can get an idea of how temperature affects its growth, and that's the idea behind variable. The experimental group, if we do like two groups, the experimental group is the group in the experiment that is identical to the control group except for one factor and is compared with the control. So I in that last slide I talked about variables. The control group is the group that you don't change anything. You don't play around with the temperature. You keep everything the same, constant, consistency, all the way through. So, and that would follow into our definition of the control group, the group in the experiment that serves as standards of comparison with another group, to which the control group is identical except for one factor. So when we talk about this identicalness except for one factor, that one factor is the variable that's changing in the experimental group or the experimental groups. Again, I want you, this is all, we're defining all these things, but we want to understand this in terms of how we would do it or apply it in an experiment. Next, we organize and analyze data. At the end of the day, as you're running your experiment, you're going to actually make more observations. The easiest way to think about this is there's going to be some type of measurements that we take. In my example with growing plants and looking for what's the optimum temperature for growth of a particular type of plant, every day you're going to come in at the same time and measure growth. You're going to measure height, maybe measure the girth or width of the stem, and those measurements that you take, that becomes data. Data is any piece of information acquired through observation or experimentation. Generally speaking, the more data points we can collect in an experiment, the better. Okay. Now that's a generalization, but generally speaking, the more the better. Okay. And then we have to do things with that data. You guys are going to see, even as you do labs, and I look for you to write these up in your lab notebook. Raw data just put down on a piece of paper, that means nothing, okay? It has very little value to it whatsoever. Data needs to be categorized. It needs to be organized, especially when we are collecting, a scientist is collecting lots of data. With computers now, we have the ability to collect almost infinite amounts of data in short periods of time. So we had better be able to organize it. Field notes are important, and every scientist has their own way and their own system for keeping field notes. They don't publish their field notes. They don't hand their field notes out. They're going to take all that stuff that's jotted down on a piece of paper and they're going to organize it. And that's where we see tables come into play in science, graphs, and if you read a research article in a journal, at some point you're going to get and find the data, the results, and you're going to find tables and graphs. And these things are basically the language that scientists speak in, because it makes it easier for us to analyze the data, analyze the experiments. Graphs are generally thought of even as better tools than tables, because graphs give us a quick, we can get the information off of a graph really quickly. Tables, smaller tables, sure, but larger tables, no. But a bar graph, it's easy for me to see which one had the most growth real quickly. So graphs become really important. We see this again about bar graphs. An example here, that bar graph comes from a table of information, but you can get that information much more quickly by looking at the bar graph than you can by reading the table. I would think by now, as you're looking at this bar graph, you know what it's all about, and you've seen it, okay? But it comes from this table, and this table is not necessarily as clear and concise and quick to read and get information off of as that bar graph was. You see them in a side-by-side comparison, how much easier it is to get the data off of the graph than it is the table. Now imagine, this is a table with three data points, or 3 sites, 6 data points total. Imagine if it was a table that had 600 data points. We get to see how a graph would be much easier. From this point, we draw conclusions. Okay, we've made observations, we've come up with a hypothesis, we've predicted, we've experimented, we've collected our data, and now we're able to analyze that data, and we determine the results. Analyzing the data, comparing the outcome with the prediction. Does the data uphold and support the prediction, or does it do the opposite regarding the prediction? And what a scientist hopes for, if their hypothesis is really tight and strong, if their experimental design is well done based on the prediction, is that from the information they've gathered from their experiment, they can form an obvious conclusion. But that's not always the case, okay? It's not always the case even after we've done all this work. And I want you guys to understand something. In research, this, getting to drawing conclusions, this isn't, we did this, we did this experiment in class, and 3 weeks down the road we got our information, our data. This might be 3 years down the road. This might be 10 years down the road. That's the pace at which research works at, and you may be 10 years in, finally you've collected all your data and you're looking, and even still your conclusion is not that obvious. And it can be a tough row to hoe, so to speak. And so computers and mathematical tools have come into play. Statistics in particular, and we don't go into statistics a lot in this class, but statistics help the scientists analyze their data more clearly and more concisely, especially when there's large amounts of data, and help them determine if their results are meaningful, if they have value, or if they're coincidental. And then a big part of this that I think gets missed a lot is that the experiments need to be repeatable. One of the things I'll say at the beginning of this class is science, at its most foundational level, is what we can observe, and it's repeatable. Good science needs to be observable and repeatable. Now, I think understanding that means that we have a bit of a tension or problem in our culture today with certain quote-unquote scientific things that are debated. Okay, we find that we have in the realm of quote-unquote science debates about things that aren't observable and aren't repeatable, right? In particular, origins, right? We have evolutionists on one side. We have intelligent designers somewhere in the middle. We have creationists on the other side. And the scientists who are evolutionists, and the scientists who are intelligent designers, and the scientists who are creationists are all debating certain things, almost seemingly as if it's science, but they're debating something that can't be observable and cannot be repeatable. And so we're going to talk about that a little bit in class, but I want you to be thinking about that. I think maybe what that means is we need to rethink our categories and how we define things and how we explain things. If a research scientist does everything we've talked about, spends 3 years, and gets their results, they're not done. They go and do it again. Or they're running the same experiment simultaneously in 2 different places. Or there's another lab halfway around the world running the same experiment. these things there's very little value in the scientific world for results that are found the first time through. But once you find those results, you publish them, and another scientist says, well, this is connected to what I'm looking at, and they're going to see if they can repeat the results that you got. Well, I'll run this test. And then another scientist says, I'll run this test. And eventually, you have a whole bunch of scientists over the course of a long period of time who have run the tests over and over and over again, have gotten the same results over and over and over again. And now, everybody believes and views that that has some appropriate weight to it. And that's important to the scientific process as well. The more often an experiment can be repeated with the same results in different places by different people, the more sure scientists become about the reliability of the conclusions. So supporting evidence is very important before a hypothesis is accepted. And scientists need to communicate their results, and so scientists publish. They publish journal articles, and it's not necessarily easy to get your work published in a journal article. These are called peer review. That is PEER. That means before it gets published, a whole bunch of other scientists read it and decide if it's good science, if the hypothesis was strong, if the conclusions were valid before it will get published. But once it gets published, then it becomes part of the common knowledge of science other scientists will go to and use it to help aid in the work that they're doing. All right, the correlation method. Now we've laid out this whole scientific method, experimental method, this whole process. What if a scientist wants to test something that they can't easily test? Maybe they can't get to it, maybe it's unethical. What scientists do then is they find something that they can test, that they can easily get to, that it is ethical for them to test, that has some type of connection to the actual thing that they're interested in. And this is called using correlations. And if I can test this thing over here that has some correlative connection to the thing I'm interested in, I can get results about that, I can probably infer some stuff about the thing I'm interested in, and that's using the correlation method. By definition, correlation is the linear dependence between two variables. So if we know that there's some dependence between variable A and variable B, and I can't test variable A, but I can test variable B, I can run my experiments and my tests on variable B and infer some things from those results about variable A. And that's the correlation method. Don't worry about the example here. Yeah, I don't spend a lot of time covering that. Correlation method has its pros and its cons. They're definitely useful, but it's very hard to make the argument if you use a correlation study to make an argument that you're showing a cause and effect relationship. Okay, and so they have their weaknesses too. They show us something. We know that if there's a good, strong correlation between variable A and variable B, we can test variable B, and it shows us something about variable A. But it's really hard to make that extra step and say it shows us cause and effect relationships. So that's a weakness. But you can look for correlations in multiple places and avenues of research, and that helps. I'm just going to go over this section briefly, especially I want you to think about this in terms of the movie Expelled that we watched. Because there we have a textbook that's telling us that good scientists, share several key habits of mind, ways of approaching things. I want you to think about this list in terms of some of the things we heard scientists saying or we saw people in power in the academic institutions doing, and I want you to draw conclusions whether or not you think all this matches up. The first, good scientist needs to be curious. Curiosity is important. Good scientists are constantly asking questions. Well, why does this happen this way? How does this work? And curiosity is really important. Second, skepticism. Okay? What do we mean when we say a good scientist is skeptic? Well, I think what the textbook is trying to get to here, and I think what we mean is that for scientists, the simplest, most obvious answer is never enough. In real life, the simplest, most obvious answer may be the most important thing out there. Okay? But scientists say, you know, if scientists ask a question and somebody has an answer, they may, well, that's great, but there's more. Or is there more? Or do I need to look for more? And so I think that is important. Or it's in the sense of, well, this is the accepted answer for this question, but it doesn't seem to really answer the question anymore based on new information. So I'm a little skeptical that, you know, we need to stop here. Maybe we need to go and dig further. So maybe think about it that way. Next, openness to new ideas. So science, at its very root, by definition, needs to be open to new ideas, new explanations. When we get new information, it helps explain things in a different way than what has been accepted. Good scientists keep an open mind to how the world works, even when a new explanation is being offered. They're open to it. Okay? Prove it or disprove it. Scientists don't have the option of dismissing something out of hand. Intellectual honesty is a habit of mind. Good scientists recognize the results of an experiment even though it does not go with their prediction. We do our experiment and it completely disproves what we've been working on for the last 10 years. Well, scientists have the option at that point of being honest about that and reporting their work or not. And unfortunately, there's a history within science where some scientists have not been honest about these types of things. And then good scientists share imagination and creativity. And I think that's really important because I think we don't talk about it and think about it a lot. When I went into college and I decided I was going to be a bio major, one of the reasons I decided to be a science major is because in high school, I really, really liked the big idea of science. I was always an idea person, and I liked thinking about the ideas and the theories. What I found was, as you got closer and closer to boots on the ground, lab bench work, I didn't necessarily have a real strong knack for that. And I was not a very creative person at all. It was never easy for me to be given a question and creatively work through what the answer might be and imaginatively try to design an experiment to show that. I struggled in those areas. The research, in particular, the research scientists that I got to know and I worked with were obviously some of the most creative people that I ever met. And the reason is because they were able to look at a question and puzzle through it and come up with an explanation and design an experiment all from scratch. And I think that's absolutely amazing. I think it's certainly a talent and a gifting that they were given. And I think it's good to think about that as good habits and good characteristics of scientists who do good work. They're open to new ideas, but they're able to come up with new ideas themselves and work them in what they do. All right, an example of this is here. And you can see how this imagination creativity plays out in this example as you take a look at these slides.